These three things, whenever we talk about the somatic recombination, junctional diversity, the combination of diversity, this is referring as before you encountering antigens, okay? We also sometimes call them as a primary immunoglobulin. It's called primary, you can say primary, all right? And I'm referring as Ig as immunoglobulin and gene rearrangement, all right? This is the first thing that's happening here, all right? Now after this, all right, you meet this receptor, the B cells, remember, it hasn't synthesized properly. So now after the pro B cell, this is what's going to happen. And this phase right here, this is going to say, I'll say this as a pre, or like you can simply say the pre B cells, all right? The pre B cells. And this is where, you know, what you're going to have, there's where you're going to start seeing expressions of this, you know, B cell receptor. But this is what? This is, I'm only drawing as what this, the heavy chains, right? You only see what heavy chains. At the same time, okay, here you also start seeing like other chains right here. And these are single transductions. Usually these are, you know, they're made out of alpha and beta, right? You also start seeing this single transaction molecule down here. And you see, like, you know, here, you see a chain, right? The chain right here, there's a light chain, but this light chain is, okay, we call them a light chain, but these are called surrogate, all right? Surrogate light chains. The reason why we call them a surrogate light chain is because these chains, you know, these are synthesized either by, all right? Because it has two portions, right? You have a variable portions and a constant portion, right? This variable portions, you know, are synthesized by something called like a pre-B, and then this portion right here, the constant region, that is synthesized by lambda 5. This is extra I'm giving it to you. You don't have to remember this, but, you know, so because of that, you know, and they are covalent, and this right now, they're covalently bound with this heavy chains. We call this light chain as a surrogate light chains, okay? And because this is like, sometimes we say this artificial, this light chains that you get to see in this pre-B cell stage, right? And you obviously have this single transduction molecule. Now, one concept that I have to talk about here is that, you know, when these B cells are going through the process of proliferation to maturation, you know, they also have to go something called checkpoint, all right? That's what they do. And this process where they go like multiple checkpoint, okay, this process, and they have to get selected to be further mature, okay, for the maturation to occur. This process is what is called your central tolerance, and this is happening in where? Central tolerance. And this is happening in the, right now, the bone marrow house. And usually when there is a central tolerance, there's three things you have to guys remember. Because it happens in three ways. Number one, there is something called receptor editing. All right? And then second thing that you have to remember here is that, all right, let me just write it on this call. There's a clonal deletions. All right, the clonal deletions is there. And the third is something called energy. What is the concept of this? Okay, when these guys are, okay, now look, these pre-B cells, you have what? You have a heavy chains, you have single transaction, right? And you also have this, the surrogate light chains, right? And they're covalently bound one another. What has happened is that, you know, during this differentiation, the B cells, let's say the B cells are given, let me make this B cells as this one. The B cells are given myself antigens. Let's say this my, all right? And let's make this as a myself antigen. And this self antigen could be like multivalent, all right? What are multivalent? You know, like your bone marrow house, there are a lot of stromal cells there. And those stromal cells are what? The adhesion molecules like ICAM, BCAMs, right? And, you know, and they have that. And on top of that, you know, this multivalent, which basically means, you know, they have this like glycoproteins, right? The proteoglycans, all of that. And these are what? These are our antigens. These are self-antigens. And what is going to happen is that whenever, you know, you are giving our self-antigen to, let's say, the B cells, if the B cells reacts it, that means this is what? This is autoreactive B cells. Okay? If it is strong, let's say, autoreactive B cells. It's a very important concept here. This is called autoreactive B cells. All right? And if And the way it does is that they do a cross link whenever they are, whenever you present self-antigens. And now this process, what they do is that they will respond to this, our self-antigens, right? And if they do auto-react, that means what is that? They have to go a process of apoptosis, right? Because we don't want that cells. We don't want that cell because that's going to hunt our own body, right? The way they do this process is that the apoptosis occurs is that, first when you present our self-antigens like multivalent, like glycoproteins or proteoglycans, what is going to happen is that they go process of something called receptor editing, meaning that this is going to cause different signal transduction molecule, okay, and they go all this gene rearrangement, all that, and then they will express, there is a difference. What is going to happen? There is going to, okay, they make a new light chains, okay? This is about the light chains. They'll make a new light chains, because this is light chains is where, like look at this light chains right here, okay? Because these are the one where, they will what? They will hold the antigens, right? So they'll make a different light chain, let's say. And if let's say this light chains reacts to the antigens and it does not, let's say, self-react. It's not if it doesn't auto-react. So this is like, now let's say these B cells, okay, we'll make different B cells, all right? So if this is new B cells and they have this other receptor right here, and if this other receptor does not react to the self-antigens, okay, mean this is a non-reactive, okay, non-autoreactive B cells. If this is a non-autoreactive B cell, this is now gets selected for the further proliferation maturations. But if it's because if it's autoreactive, then remember, again let's say this guy become again, even after going self-editing, doing the rearrangement of the genes, it's still, it encoded, let's say, another gene or another receptor right here, okay, that is autoreactive to this antigens, okay, and they will keep doing that, okay, they'll keep rearranging the gene until they can actually find that receptor that does not react to antigens, all right. And there is eventually what happens is that, they run out of these genes, all right? They run out of this, let's say, the light chains. So if they run out of the light chains, what is going to happen is that these cells have to go process of apoptosis, okay? And the process of these cells go apoptosis is right here. This is what is called clonal deletions, all right? So remember, the central tolerance happening in 3 ways. Multivalent proteins, all right? Whenever you are presented to our B cells, right? These B cells, if these are autoreactive B cells, right? Then what is going to happen here is that they'll auto-react and then they'll accept it, they'll do rearrangement of the gene, doing something called receptor editing, and they'll express a new set of this light chains, right? Which can actually work on the antigens. But even when you make a new, let's say, light chains and still, it is autoreactive, all right? They'll continue doing receptor editing until they run out of this, this different sequence which actually gonna make the light chains, then they'll go process of apoptosis, all right? And that process is what is called clonal deletions, right? And this is how, all right? Very, very early on, even in the bone marrow house, the B cells are taught not to react on our own cells, our own antigens. Because if the B cells are starting reacting to our own cells, then that will lead into autoimmune disease, all right? Now I also have to talk about this. What is the concept of this energy thing, okay? This energy is basically what it means is that usually these response to the soluble proteins, or let's say this is multi, right? So monovalent proteins, right? So whenever, let's say, monovalent proteins are attached to these B cells, okay, we'll make this another B cells right here, and whenever, let's say, there's a B cells and monovalent proteins are right here, okay, then what is going to happen here is that, this IgM will take inside this, right, do a different signal transductions, and then what is going to happen is that they'll lead into expressions of a lot of these molecules, okay, and maybe these are not IgM, but these are like IgD, IgD, all right? IgD, a lot of IgD, and the less IgM. And whenever that happens, these IgDs are very, very weak, weak responders of the antigens, right? And whenever there's a weak antigens, they'll either they'll no respond, right? Or there will not be, or maybe weak signal transduction will occur, okay? The process in which, they don't respond to the antigens or very weak respond to that antigen, that is what is called anergy. Remember, you think about energy, meaning that they did not use energy to synthesize or to respond to that antigen. You can think about that way, okay? And this is usually, these three concepts, we call them as a central tolerance process, right? This is what is happening. And this is how the selection process takes place, right? I know I'm combining both this concept of how the gene gets rearrangement and also selection process, because this is how, we should be thinking about it too, because this is how you're able to understand better, all right? Because because as we proliferate, as this B cell proliferate, at the same time, there's also, this central tolerance also taking place, all right? The rearrangement genes happening and also central tolerance also happening here, right? Now, after this concept right here, right? This previous cell now, come back here. The previous cells you have this receptor right here, all right? And then they have a single transduction molecule. Now, this is usually they refer as a large pre-B cells, all right? And this is going to be a, let's say, small pre-B cell. In the small pre-B cells, or, what is going to happen, or immature B cells, well, let's just say, what is going to happen is that, they will synthesize here more. This is going to be my heavy chain. But remember, guys, this is a heavy chain right here, right? And obviously, this heavy chain has a light chain, gamma, and concentration, and all that stuff. And then they'll make this, let's say, this guy, and this is our maybe a kappa chain, all right? And this is probably our receptor right here. All right. Now, and then they also have this transduction molecule right here. All right. Now let's say this is like a immature after a let's say large. The concept here is that there's going to be large pre-B cells. Okay, and then that will lead into the formations of small B cell. That's where you're going to start seeing the whole this, your light chains. Also, light chains, which are not surrogate light chains, but proper light chains, okay, and they're covalently bound with they are bind with one another with, your sulfide bond, disulfide bond, right? They bound one another with the disulfide bond. If you look at this structure right here, look at this one right here. This is what is called disulfide bond, right? SSAH. And this is also in light chains, heavy chains are bound with a single sulfide bond, right? Disulfide bond. That's what is going to happen here. Same thing here. Now, once there is, let's say, the receptors are formed, right? This receptor, we call them a BCR receptors, all right? BCR receptor. Or they sometimes they refer as because it looks like IgM, is an IgM receptor, okay? This is where I need to make sure that you understand this here. This is an IgM receptor or BCR receptor. Whenever you think of a receptor, this has to bound with what? Plasma membrane, all right? This IgM that is bound with this plasma membrane, this is not an antibodies, because antibodies are what? These are what? Why, we often say antibodies are what? Humoral. What is humoral? What is the concept of humoral here? Because these B cells are humoral mediated immune system, right? HMIS, right? Humoral. Whenever we think of humoral, let's say humoral, right? Think about fluid, right? Because they're going to float around, all right? So antibodies, they are not bound with the plasma membranes. They freely move around into your extracellular fluid. They flow around from your blood to your tissues, all right? Extracellularly. But these IgMs are bound here. And what is the difference between let's say this IgM, which is an antibody, let's say, and then IgM receptor? The difference is the C terminus, like here, right here, at the C terminus. At the C terminus, what happens is that there is a hydrophobic amino acid chain attached into this receptor, okay? If there's a hydrophobic amino acid is attached to this C terminus, That means your plasma membranes are hydrophobic, so because they will strongly bound one another. But if you look at the IgM antibodies, the C terminus, the hydrophobic is missing there, right? Instead of that, there's a hydrophilic portion attached to that. That is the only difference between IgM antibodies and IgM receptor. It is the C terminus, okay, where there's a hydrophobicity is there when it comes to the receptor. And hydrophobicity is provided by like poly A tail. And those are usually hydrophobic, all right? Now, after the little difference about that, let's come back here. After this, right, once you have formations of this, right, there's one more concept that you have to remember here. In this phase, right, before, like now they have IgM receptor. They have receptor, there's signal transition molecules, all of them there. But still, this is not a mature B cells. We call this as immature B cells, all right? This is immature B cells. At the same time, these pro B cells have a CD10 right here. This is also have CD10 right here. They also have a CD19 right here, right? They have this. What is going to happen here is that when you get from the previous, when you get differentiated into the, let's say, immature B cells, right? They are going to gain another, like this guy right here. CD19 is going to be there. They will gain another protein which is called CD20. There's also what? CD10 is there, right? This is immature B cells. Now, one concept that I have to talk about here is that whenever, let's say, when these pre-B cells, when you get differentiated into, let's say, immature B cells, right? During this process, right? This is where, you'll see something called a checkpoint. And what is that concept of a checkpoint? Let me explain this concept right here, all right? So, if you look at this right here, if I make this structure right here, all right? Let's say there's a B cell, all right? And there's a receptor right here, obviously, right? Obviously heavy chains, and you're gonna have let's say there's a light chains right here, all right? Whenever there's a, let's say with the single transductions, there's single transition molecules here, all right? And they have this lot along these short cytoplasmic tails. We usually call them, this portion right is called ITAM, right? Now, what is going to happen here is that this lead into, okay, when they put this, let's say the BCR receptor is an antigen, that's going to get, that's going to lead to activations of this family of the tyrosine. We usually refer as SYK, all right? This belong to the serine family, this belongs to tyrosine family, right? So the protein kinase family, okay, sometimes they refer as a spleen. Now, when they are in immature B cells, as we discussed, they have in immature B cells, they have CD19, CD20, CD10 markers, and now you have like BCR receptor, you have with proper BCR receptor, and these are selected to move forward, right? Again, still this immature B cell though, right? Now, when they go to, let's say, blood, now they're gonna go migrate to the blood and their peripheral organs. So this is a blood right here, and this is I'm making as one lymph node, or it could be, let's say, spleen for our case, okay? And spleen, if you remember, this is what we call the cortex area. There's a paracortex area, right? All of them. And usually in the cortex area, that is where the B cells go and get marginated, all right? Now, when they go to, let's say, blood, okay? When they go to the peripheral organ, here what is going to happen is that, in the blood also, they're going to also, respond to like different antigens, all right? And they have only IgM so far, right? And what is going to happen here is that this guy, let's refer as this as IgM right here, what is going to happen is that this guy as well, they'll take antigens and, some of the genes inside here, they'll start, synthesizing some other type of receptors, and you see this IgD, all right? IgM and IgD. So, now this becomes mature B cells when you have both IgM and IgD, all right? But remember, these B cells, even though they're matured and they express both IgM and IgD, but they are not, they are not active yet. They're not effector B cells yet, all right? These guys are naive B cells, all right? These guys are naive B cells. And even they will go in this right here in the, let's say in the cortex area, all right? Specifically in the cortex area, they go to primary follicles area, and then after that, what happens is when they get exposed to the antigens, then they will, okay, they will get differentiated, all right, into plasma cells, and they'll start making antibodies, right? That process where they go all this, the like isotypic switching, or are making into like different types of like antibodies, that process, okay, that usually takes place in the cortex area, but especially the cortex area, there's something called germinal center. Germinal center, right? That is where, these B cells, let's say maturations and isotypic switching, all of them take place. And remember, that only happens, right? But that happens when they encounter the antigens. B cells gets activated, all right? And this isotypic determines the switching that happens, that takes place after the, what is this called? After, what do we call this? After they encounter the antigens. This process so far we talked about right here, right? So far right here, this process, the mature, naive, this process, all this is before they encountering antigens, right? And the three things that happen that took a very, very important role. We talked about the somatic rearrangement of the gene, right? We call them somatic recombinations. We talk about junctional diversity, right? At the different junction, at the heavy chains, okay? They are doing alternative splicing to make either IgM, IgA, IgD, right? And then third is they also did what? There's a concept called, they combine the heavy and light chains, okay? That is what it's called combinatorial diversities happen. Those are three things happen. And that happens, right? And they also went a process, selection process here too. They went a selection process here, which is called central tolerance, right? Receptor anergy, clonal deletions, and energy that also, took a place in this time, all right? Because remember, one thing that we have to talk about is how they got developed, right? They were born, and then how they were started what? Maturing, okay? And during the maturation, the central tolerance also takes place because our cells should not react to our B cells, okay? Our immune cells should not be reacting to our own antigens. Because if that happens, then that is what is called autoimmune disease, right? So this is why in a very, very early phase, right, this maturation was taking place in the B cells, or in the bone marrow house. That's why, we call this process, okay, like up to here, the naive B cells which are not activated, they were matured here, okay, but they were mature, but they were just not activated yet, right? So this was taking place here in the bone marrow house.